Hello, everybody. I'll be talking about the role of two spin-out biotech companies from our research. The first one, Fibrotech, the second one, Fibrocor, really looking at the role of fibrosis in multiple organs with focusing in this talk in particular on the role of fibrosis and therapies against it for heart failure. Here are my disclosures. I work with major pharmaceutical companies. I'm founder and shareholder of a couple of biotech companies. First one is Fibrotech Therapeutics and Ocurix, and then more recently I am the co-founder, shareholder, and chief scientific officer of Fibrocor Therapeutics. I owe a lot of my understanding of heart failure and cardiac disease to my former PhD student and now close colleague and collaborator, Dr. Kim Connolly, who's a clinician scientist here at St. Michael's Hospital. I've known him, as you can tell from the photographs, for quite a long time now. So the heart we normally think of as being composed of contracting cardiac myocytes and really ignore the fibrotic component or the extracellular matrix, but it surrounds the cardiac myocytes, it surrounds the bundles of cardiac myocytes, and really has a critical role in the establishment of adequate contraction and relaxation. So there are collagen bands that connect individual myocytes that run between them, and their ability to connect is very important for the contractile apparatus to work in a coordinated fashion. On the bottom, you can see that there are these sort of tendons of fibrotic material that go between individual cardiac myocytes and cardiac myocyte bundles. And when those areas of fibrous tissue of collagen become disordered in cardiac disease, there's misalignment, and cardiac contractility and relaxation become significantly impaired. So there is what we call replacement fibrosis, where large areas of cardiac myocytes are actually replaced by fibrotic material. There's interstitial fibrosis, where the normal paper-thin areas of extracellular matrix between cardiac myocytes and bundles are replaced by thick collagen bands that interfere with contraction. And then there's also perivascular fibrosis, with fibrosis around vessels that interferes with vascular function. If we look at heart disease in diabetes, for instance, these are patients with heart failure due to what we think is diabetic cardiomyopathy. They don't have coronary artery disease at all, but they have heart failure. We see the extracellular matrix deposition in blue on this trichrome stained section is between cardiac myocytes that you can see, and the extent of the interstitial fibrosis and replacement fibrosis is increased by diabetes itself, but increased even further in patients who have diabetes and hypertension. If we look at patients with heart failure with reduced ejection fraction, there is a very close relationship between the percent of the cardiac tissue that's occupied by collagen volume fraction. So this is the extracellular matrix fraction on the horizontal axis, and the left ventricular end diastolic pressure on the vertical axis. So the more fibrosis, the worse the cardiac function. And on the right, we see that patients who have severe fibrosis have a far worse prognosis than patients who have mild fibrosis. That's heart failure with reduced ejection fraction. Same happens with HFpEF, with preserved ejection fraction. When there is increased collagen, then there's evidence of increasing diastolic dysfunction, as evidenced by the E over E prime that we see on the horizontal axis, and the type 1 collagen volume fraction on the vertical axis. So the more collagen, the less functional activity we have in the heart, both in terms of contractility and the ability to relax in diastole. We see it in lung tissue as well, where there's fibrosis. And here we have an antifibrotic drug called pirfenidone that actually is in clinical use that reduces interstitial fibrosis in patients with idiopathic pulmonary fibrosis. Its trade name is Esbriet. We have one study to show that pirfenidone was actually effective in reducing cardiac collagen in patients with HFpEF in this recently reported randomized controlled trial, phase 2, so relatively small trial that was published only 3 months ago in Nature Medicine. So the whole idea of being able to treat fibrosis as a way of introducing a novel type of therapy to heart failure is up and running. Our task and what our laboratory has been interested in is, can we develop new drugs that treat fibrosis? And as you know, with laboratory research at universities like we do, we're really at the sort of the center of trying to discover things, but we really can't get things into therapeutic use because we don't have the funds or the technology to actually invent a therapy that can go into human disease. And so this is what we commonly call the research valley of death, where we're unable to get our laboratory research into the clinic. We were very fortunate about now 7-8 years ago or so in getting a $3 million grant from the NIH, specifically that was designed to take bench research into the clinical first-in-human studies. So here are my colleagues from the first company that we span out, a company called Fibrotech Therapeutics. Darren Kelly, who was a postdoc in my lab and then became a principal investigator, my good friend and colleague Henry Crum, who unfortunately died not so long ago, and myself looking much younger than I do now. And we formed a biotech company and developed a drug that inhibited fibrosis, and we tried it in the heart. So here is a series of chemical compounds. The compound that we progressed into clinical trial was one called FT11. It is a drug that inhibits transforming growth factor beta-induced fibrogenesis and PDGF-induced fibroblast proliferation. And you can see that there on the top right, there is dose-dependent reduction in the synthesis of collagen with our compound FT11. And on the bottom right is cell proliferation in fibroblasts, dose-dependent reduction in fibroblast proliferation with the drug. Looked good in vitro. We then took it in vivo and showed that we could reduce the deposition of type 3 collagen in post-myocardial infarction, adverse cardiac remodeling, fibrosis. And we were able to look at that histologically. And if you look at G there on the right panel with the photomicrographs, you can see type 3 collagen immunostaining in brown with in the post-myocardial infarction remote zone. And when we give our drug FT11, where there's a substantial diminution in fibrosis. And when we look at cardiac function with echocardiography, in the rats that had myocardial infarction and then vehicle treatment, their fractional shortening progressively decreased, whereas the animals that got our antifibrotic compound had preserved fractional shortening and ejection fraction. And we wanted to be able to prove that it wasn't just in the post-myocardial infarction, that other causes of cardiac fibrosis would also be ameliorated by our treatment. So here is a model of diabetic cardiomyopathy, where we look and see that with this picrocereus red staining in panel C, you see that there is interstitial fibrosis as evidenced by the red staining with picrocereus red, with a substantial diminution when we give our drug FT11. And the cardiac indices, when we do pressure volume loop acquisition of cardiac function, we see improvement in the in passive diastole, as the end diastolic pressure volume relationship. We don't see much of a change with tau, but we do with the preload recruitable stroke work, indicating that there's improvement in systolic function. So with that background, we went into a human clinical safety study in 2014 that showed no adverse events. And then the company was essentially gobbled up quickly and acquired by a major pharmaceutical company called Shire. So we're back at then, having sold the company, back at the laboratory trying to investigate new therapies once more. And here we applied with colleagues Darren Ewan and Jeff Ranner to the Banting and Best Diabetes Centre transformational grant application that they had to develop new antifibrotic therapies, crossing the valley of death again. $1 million that grant was for was not enough. So we were very fortunate in being able to create a sense of enthusiasm around our spin out, and managed to cobble together another couple of million that launched us into being able to form a company. So I'm the CSO, Mark Steedman is our CEO, Bill Fanslow is an antibody expert who's the VP of drug development, and we have a chief financial officer, Joe Wallowitz, as well. So with that, we've actually launched 3 programs. One of them, 4586, is due to first in humans as an antifibrotic in the first quarter of next year. We have another series, 918, that is going into investigational new drug development, which is the prelude to human studies in the first quarter of next year, and then one following not so far behind in the fourth quarter of next year. So it's still a long way to go to get the drug to phase 2 and then phase 3 and then onto the market with up to several tens, if not hundreds of millions of dollars. But at least we're on the way. We think we've crossed the funding valley of death and now on the uphill march to try and get what we believe are new therapies for fibrosis in general, but specifically would be a new therapeutic strategy in heart failure. Thank you.